a bioinformatician and also theoretical. He worked before here, maybe his face is familiar to you, as a bioinformatician also. And during his career, he studies working in genetics, and then mostly in bioinformatics with genomics. Now he owns his company, is a CEO in his company, Omics Bioinformatics, and today he's gonna talk about mysteries of virus hosts, and I think it's bats. No? Thank you very much for coming. So the word is yours.
Presentation. Okay. So this is the title of my talk of today, Revealing the Mystery of Virus-Host Interaction Through Bioinformatics. This is more or less the work, I mean, partially the work that I did during a postdoc at Mount Sinai between December 2020 and December 2023. Mount Sinai is an hospital at New York City. This is the Mount Sinai Central, and I was working here in the Annenberg building. Thank you, doing a postdoc. Thank you to Professor Adolfo García-Sastre. He is a very important expert in flu virus. When started the COVID pandemic here in Spain, I was thinking in apply all my knowledge to virus because I was very interested in apply all my background to virology. So I contacted to Adolfo, and he gave me this important opportunity. And this is the work that I have published, sent to my work in Mount Sinai. This is a paper from Cell. You know that Cell has an impact factor of 60. I am the second author of this paper. This is other paper. I am in the middle of the author. This is a very important journal in the virology. Thanks to the partially of the work that I did in this paper, speaking with Lisa Miorin, that is the corresponding author, one of the corresponding author of this paper, and thanks to Adolfo too, we use part of my work in collaboration with JJ Fernandez and other collaborators to apply some of this data to other papers. This is Biochemica, it's a Biophysics acta that has an impact factor of 7, something like this. Okay, today I will speak about the first paper that I have from this postdoc, and a little bit about one of the work that I did with Minayon, that is a device in order to take sequences but with long reads. Okay, this is all the author of the paper. In this paper, there was a very important collaboration from Javier Juste and Carlos Ibáñez from here, from the Doñana Biological Station, because they provide a sample from the bat that we are very interested to do this research. I know because this morning I was speaking with Javier and Carlos that send this sample in the middle of the pandemic to Mount Sinai was very difficult. So I really appreciate the work that all the people do during this period in order to send the sample to Mount Sinai. So the COVID pandemic, more or less in number, you know that have more or less 7 million of confirmed deaths. We have more or less 700 million of confirmed cases, and this is the vaccine doses administered at today. So the question is, at the beginning of the pandemic appeared several studies. One of more important is this, that point that maybe the virus, the SARS-CoV-2, have an origin in a host that is a bat species, okay? And the closest, at this time, the closest no virus to SARS-CoV-2 was this rats 13. And this virus, the host of this virus, is this bat, Rhinolophus affinis, from the Rhinophidae family. So Adolfo García-Sastre have the idea to obtain samples from this family in order to study the infection of this virus in this host, because there is a lot of virus that can infect bat, but apparently the bats don't have illness for this infection. So the idea is to do a comparison between the infection in the bats and infection in the human cells. So the question is, do the bats of Rhinophidae family have something special in their immune system that allow them to deal with this kind of virus? Okay, this is the Rhinolophus ferrumecinum bat, and this is the range of the bat, that is from Japan to Spain, more or less. Okay, and the sample of Rhinolophus ferrumecinum that we use for our study was collected in Jerez de la Frontera in Spain, more or less here, from thanks to Javier Juste and Carlos Ibáñez, and was sent in the middle of the pandemic to Mount Sinai. And in this study, we use other bat, that is this species, Myotis myotis, and this is the range of this species of bat. This sample of Myotis myotis was collected here in France, in the Morbihan, from thanks to the Dr. Emma Zetteling's group, a member of British Vivanti, local volunteer, and a student from University College of Dublin. So one important point in order to study the infection that produce a virus to a host is obtain cells. In order to work with this cell, we need that this cell was immortalized or get a stem cell. So in the Thomas Awaka lab, they did a protocol in order to obtain that we know bat-induced pluripotent stem cell. They started with embryonic fibroblast isolated because the sample that was sent to Mount Sinai from the Rhinolophus ferrumecinum was a female and was pregnant. So fortunately, they can take embryonic fibroblasts and isolate it and apply a Yamanaka protocol, modify Yamanaka protocol in order to obtain the bit cell. Okay, in order to check that this bit cell is real stem cell, you can see here the bit cell, and this is fibroblast that is in the medium. Okay, this is other photography from the bit cell. And in order to check if these cells are real stem cell, we need to do search in order to look at the expression of genes that is related to the pluripotency of this cell. Okay, so we did an RNA-seq study in order to detect different genes that is related to the establishment and maintenance of the pluripotent ability of the cells. Okay, in order to check more in detail this, in Sawaka lab they did a study of the different layers of the that produce the stem cell, that can produce the stem cell, the ectoderm, the mesoderm, and endoderm. And they did immune staining using different markers for the different layers. This is another example because this cell in some circumstances can do a blastoid that is similar to a natural blastoid, and they mark this gene that is a very important gene to the pluripotency of the cell, OCT4. Okay, looking in more detail in this cell, in Sawaka lab, they see that appears some structure that is similar to a vesicle, and is appear to fill it, that this vesicle is filled with a membrane structure. And seeing in more detail this, in Sawaka lab, they see that there is some structure that is similar to a capsid of virus. So, join this with the literature, they find that there is a paper in Nature that point into relation the expression of endogenous retrovirus with different states of the embryo. Okay, at some point of the development of the embryo, in the human embryo, there is an activation of the expression of this endogenous retrovirus. So they think that probably there is a relation between the two things, so probably this B cell is producing virus. They check this with Western blotting using a special antibody, a specific antibody to capsid protein, and compare this with different line cells. And you can see here the case of the beet cell. Okay, we have different cells in our study. This is the Rhinopus ferromechinum fibroblast, that is the original fibroblast from the embryo. This is the beet cell, that is the pluripotent stem cell from the embryo that after apply the protocol, and this is the cell from the meiosis. You can see here that in the two species, when we study with an immunostaining the fibroblast, we don't see something that point out the detection of endogenous retrovirus, but in the cases of the stem cell, we appear, it's appear a signal that point that there is an expression of endogenous retrovirus. In order to check this more in detail, we check for different genes that are relevant to retrovirus, that is the M, the GAF, and the GAF genes, and compare the expression between two lines of beet cell against to the best cell, that is fibroblast cells. Okay, and you can see here that the expression of these genes is greater in the stem cell than in the fibroblasts. Okay. In order to check that what happened with this hypothetical virus, I did an in-depth analysis of this data, applying a protocol from metagenomic. Okay, We have Illumina sequencing reads, that is pyrene, more or less 100 base pairs, and we have 6 samples, 2 biological samples with 3 replicates A sample. Starting with this, I apply a trimming and filtering of the reads. This is a brief fly protocol, not in detail. Okay. Next, checking the quality of the reads and apply this tool. It's a specific tool for metagenomics. Okay. The problem that we have with this data is that we have around 500 million of reads in total. So the best tools in order to analyze this data is use BLAST, but BLAST takes months in order to analyze this. We are very busy in order to do this. Okay, so checking the literature, I find this software Kraken2, that is a special software from metagenomics. At the date that I use this software, it has around 600 citations, but today it has this number of citations. And this software don't work the same thing that BLAST, but it's very fast. Usually give very good result. Okay, so how works Kraken2? The first step is make a special database. Imagine that we have different sequences that we are interested in share in our data. This is the reference. So suppose we have a virus genome. This is the virus genome. We need to make a database with this virus genome. This database is composed with different cameras. Okay. By default, the size of the camera is this, 35. What is a camera? Imagine that the starting of the sequence is this, and suppose that this is the 35 sequence, is from here to here. The next camera is this, removing the adenine, and the first adenine, and adding this adenine. Okay, so every time we have 35 per base, but we move one nucleotide each time in order to cover all along the genome. So we need to do this for all the genome that we are interested in. In the case we are interested in all of the genome of the virus, so I take a special database for virus and construct the camera for all of the species of the virus and make the database. This can take maybe 2 or 3 hours for all of the virus that is in the database that is near to all, close to all. Okay. The next step, when we have the database, is do a search against our data. Okay. Imagine that we have different reads from our data, and we have different cameras. Kraken2 do something similar to an alignment. Is an exact match between the sequence. We have the read 1, and this is the one camera belong to species 1, for example. This is exactly the alignment. So all of the sequence is the same. Okay. For other reads, we have a match with other species, and in some cases, we can obtain a match between the reads and 2 different species. In this case, in order to assign a species for this read, we use the common ancestor of these two species, okay? Okay, applying this protocol, we obtain this number of virus, 2,500 virus species in all of our data, and this virus species belong to close to all group of virus. So this is something strange, and in order to check this, we did a different Venn diagram in order to compare the best cell with the best cell with human, obtain that in the best cell we detect more virus with this protocol than in human. In best cell we obtain more virus than in the best cell and than in the human cell. Okay. So one of the problems is our reads are short. And maybe some of the problem that we have is that it's not this read is not enough the length that we need to do this analysis. So we did other analysis, applied the same protocol, but using other sequencing technology that is ISO-seq. This is a technology from the company PacBio. This technology produces more longer reads, so we can obtain more accurate result applying this protocol. And in this case, we have less virus. We have 356 virus, and some of the virus is shared between the original data and the ISO-seq data. Okay. So in order to do a better analysis of our original data from Illumina, I propose to apply different protocol, taking all of the read that is assigned to one species from Kraken2 and do an assembly with a software, the name is Trinity. And when we obtain the different content with this software, we can do a blast because we have less data, checking the result. So this data is more robust than the original pipeline. In order to apply this, we use a very extensive protocol, taking from one part, from one hand, taking the result from Kraken2, and from the other hand, taking the result from Premonition, that is a pipeline from the Microsoft company, and we apply this protocol from the previous slide. When we obtain the different contents of the different virus, we check the content in different special database from all the virus database, from a special mammalian virus database, non-mammalian virus database, and look at this identified virus inside the genome, back to the genome. Okay. So this is the number that we obtain. These final results are more longer by far than the original result, and this is the virus obtained in the different protocol, the number of virus, of different species of virus. So the conclusion of this work is we have obtained this cell from Rhilopus ferrumeckinum and Myotis myotis. This cell could be used in future virus infection. I know that the people from the Adolfo lab and other people from Mount Sinai, I think here, is working in this. And the retrovirus transcription has been demonstrated in this cell, and some of these viruses have been identified in detail. One of the important points that is in the conclusion of the paper is that maybe this result is related to a special immune system from the bat. That is something similar to CRISPR system in bacteria that taking a small fragment from virus and insert this small fragment in the genome, use this like self-vaccine. Okay, so this is all of the people that collaborate in this study and the entities. From the Adolfo García-Sastre lab, from the Thomas Aguaca lab, the people from here, from the Doñana Biological Station, the people from Emma Zetelin. The referees for this paper was Thomas, Adolfo, and Emma Telling. This is the two people from the Microsoft, Premonition. This is other lab from the Mount Sinai. This is a company. This is an important scientific with a very important background in genomics. This is the people from the Goethe University, and this is other entities funding the research. And the other thing that I was interested in telling today is one of my work in the Adolfo García-Sastre lab was to develop the pipeline in order to analyze the data that produce this machine, Minayon. Okay, this is the Minayon. It's a small device that can sequence long reads from DNA and directly from RNA. Okay, how this device works? It has a special chip. The name is ICTS-Doñana. This ICTS has a sensor chip, and in the sensor chip it has a membrane and a special pore. This protein, this pore, can be selective used in order to pass through this pore DNA or RNA. This is different machine that this company have, that Oxford Nanopore have, with different performance. But in the lab of Adolfo, the people use this device. Okay, this is how this device works. And a special protein carry the DNA or RNA to the pore, and the DNA or RNA pass through the pore. And this system can, because the different nucleated have different charge, this system can detect the difference between the charge, so we can know what is the base that is passing in every time through this system. So what is the advantage of this system? It can sequence both DNA and RNA. In this case, small transcriptome. The reads produced by the system are longer than other systems. The company claimed that up to 4 megabases. And this is very important point because when we need to do an assembly, the assembly takes time. If we have a small fragment, the assembly takes more time, and this is usually we have some error because for this process of assembly. Okay, so if we can reduce the assembly, it's better. If we can reduce the process of assembly, we can obtain more right result. And the price of this device is more or less cheap. This is today the price, and in this starter package we have 2 flows and a kit to prepare libraries. And the process from preparing the libraries to have the analysis the data is faster and does not depend on the external facility. So in the Adolfo García-Sastre lab, the people use this directly. So they don't send the sample to an external facility. And usually they obtain the sequence in 1 day or 2 days. Okay, this is an example of the final result that we have. This is different reads that we obtain. This is an example from a virus, an herpesvirus. You can see here different lines with different color. These point out different mutation, different SNP. And here we can see an indel. Sorry, an indel. And this is an insertion. So sometimes we have an error. But if we can see the alignment between a reference genome, we can check the error because usually these errors are random. For example, probably this is an error, this is an error, this is an error, this is an error, but probably this is a real mutation. Okay, so we can check this kind of error using this alignment. So in the Adolfo García-Sastre lab, they are very interested in using this technology in order to sequence flu virus. This is a picture of a diagram of flu virus. This is the different segment of the genome of the virus. So in order to sequence different segments of the virus, we need to use a primer for the different primer for the different segment. In this case, the primer is more or less the same because at the beginning of the sequence is very similar. With this technology, we can sequence different samples at the same time. So I think today we can use 12 barcodes in order to sequence up to 12 different samples. Next of this, we need to do the multiplexing. The analysis of this data is not easy, but it's possible. Okay. One of the points that I am very interested in order to do this pipeline is to check for different mutation, because okay, if you can see here the different segment of the genome of the virus is more or less similar to a chromosome. Okay, it's not exactly a chromosome, of course, but it's similar, biologically similar to a chromosome. So I expected that if this is similar to chromosome, it's probably that we can obtain mutation that is related to a chromosome, like inversion, deletion, insertion, and so on, transposition, and so on. So the problem is to check this in the data is difficult. So we need to do a special tools in order to check this. And in order to develop this tool, I developed a pipeline. So imagine that we have the problem that if we are interested in check a pipeline that only detects this kind of mutation, punctual mutation, we can check our pipeline, compare the result with other sequencing technology like Illumina or something like this, and using a sample that we know what mutation have. But the problem is we are interested in mutation that we don't know. So we need to construct a pipeline in order to check our software. So imagine that we have a reference genome, and we are detecting, different real data from flu virus, for example, and we are interested in see if our pipeline can detect insertion in our genome. Okay, so we can remove some location from our reference genomes and do apply our pipeline with the data that is supposed that don't have this deletion. Okay, so it's like we are detecting an insertion. Okay, and we can apply this protocol for every kind of chromosomic mutation. So suppose we are interested in a deletion, we can put on random sequencing or special sequencing in our reference genome and align with normal data and see if this data is mapping against this reference genome. So this is a way to detect if our pipeline can detect the deletion in a hypothetical genome. So this is the people that we working in this project. Of course, Adolfo García-Sastre, Carlet, that is the person that offered me to work in this project. Nacho Mena, that do some of the sequencing, and Sarah El Fahed, that do a lot of sequencing from the flu virus, working with me in order to develop the pipeline. That is all.
Thank you very much, Arturo. So very interesting. Now it is time for questions. Any questions show up? Here, two.
Arturo, okay, so working? Thank you very much. It was great collaborating with you with this research. And do you remember there was a big difference in the virome diversity that we found in these two species, in Myotis and Rhinolophus? I don't recall now.
You mean the number of the species of the kind of the species?
In the diversity of the retroviruses.
No.
There was no difference between them.
I'm not sure, but I think no. And most of the virus that with the last protocol, most of the virus was retrovirus.
Right.
Yeah.
And do you have any idea if there is any functionality or what they do? These retroviruses, there are these species there?
Yeah, what's on the point is, you mean if this is useful for the bat or something?
Yeah, if this has any functionality at any moment during the lifetime of the bat.
Yeah, maybe, but I think we don't have a strong proof of this, but maybe it's something related to this, something similar to this, something similar to CRISPR-Cas, or something similar to self-RNA vaccination, something like this.
Right.
For example,
do you expect this for this the very small, these very short pieces with less than 100 BPs and fragments, but not this big fragment that were able to activate as a proper virus at some point of the protocol, which was kind of surprising. No? That we were talking about that before. That in fact, in the very beginning, they thought that there were contamination. That was, they couldn't believe they were part of their own bat genome.
Yeah, maybe. I don't know, but maybe there is a side effect. Maybe the long reads. The long segment from the virus is taking from this hypothetical system that is the same system that takes the short fragment and put inside the genome of the bat. I don't know. It's a possibility, but I don't know. I think that, I mean, I hope that this virus are not completely functionality because, okay, yeah. I hope that this virus, I hope, is not completely functional because if this virus is completely functionality and can infect cells, we have a problem. Because maybe insert a virus in the genome of these bats. I mean, it's supposed that close to all of these viruses are retrovirus, okay? But maybe there is other virus. And the problem is if this system is real in the bats, maybe some virus can insert in the genome of the bats, can accumulate mutation a long time, and produce new virus particles, and this virus can infect bats or other species of animal. So, yeah.
Thank you very much.
In the first part of your talk, do you use some kind of null model to quantify the likelihood for any of your reads to match the reference genome of any virus species just by chance? For example, you have used sequences of 35 base pairs, right?
Sorry.
In that Kraken, I think is the technique,
yeah.
Right? So I can build many random sequences of 35 base pairs.
Yeah.
So what is the likelihood for any of those reads, random reads, to match any?
I don't check the likelihood, but when I see this result, for me at the beginning was very strange, and I check in detail the algorithm of the Kraken 2. One possibility, because Kraken 2 don't do an alignment, is it uses a hash. Okay, it's complicated to explain here, but when we apply this kind of technology, a hash search, we can use several errors, depending on the type of hash. One of the errors is collision. The name is collision. That you have a random sequencing that you are proposing, a random sequencing, and this random sequencing matches by chain with, not by chain, but by error, match with an hypothetical virus from the, well, no hypothetical virus, from a virus from the database, okay? But I check this, and the collision of the Kraken 2 is very, very low. I did a little script in order to make random sequencing of the 35 length and check against BLAST and check again the Kraken 2. When you construct a very long, no, it's not, you don't need a very long read indeed, but if you construct, you can prove this. If you make a random sequencing of 35 nucleotides, very, very probably you don't find this sequence in the BLAST database, in the old BLAST database. So if you have a match with Kraken 2, there is an error. Okay, I checked this, these errors are very, very, very low.
So it's very unlikely to match any random sequence that you can build
it's very,
any genome.
I mean, my result point that is very, very unlikely.
It would be more difficult the longer the sequence you consider, the more unlikely it is to find a match.
Yeah, this is another possibility in order to improve the result, because I use this with the default camera size, but you can increase the camera size. But at the end, you have the problem that you don't have all the virus. So you need to do an assembly. At some point, you need to do an assembly and look at again inside the genome of the bats and check if this genome is a real genome inside the genome of the bat. Yeah.
Okay.
Thanks, Arturo, for your talk. I understand you have been focused a lot on improving tools for detecting virus and classifying virus in species. So, but once you have these improved tools, do you have any plans to apply those in a research project? And what kind of things you think could be better addressed with these tools?
In order to do a metagenomic analysis of this kind of analysis, take time. First of all, because one of the problems that we have is that we are very busy. So we can analyze this data with BLAST, but take several months, maybe 2 or 3 months, and we don't have time at this point.
No, I mean, but once you have the new fast and improved tools, what kind of problems you think it could be applied to? What kind of questions you think are now easier to respond generally in research?
But like an objective,
yes.
Like a final objective?
Yes.
Okay. Look at if these viruses are real virus, but you need to do because okay, I am my background is a bioinformatician, but I know that you can see different sequences, but you can to prove this. So the only way to prove this, if you are saying that there is a virus, it take this sequence and infect cell. This take time, but it's important to do. Okay. One of the problems, I think, for example, I think 2 years from the beginning of the pandemic, some people point out that is a virus that is similar to SARS-CoV-2, and they show the sequence. The project is BAL. The name of the virus is BAL something because the project is BAL something. I don't remember the number. But the virus, they don't prove that this virus can infect cells. So this is an important point in order to check this kind of thing.
Okay, thanks.
Okay, any more? Okay, so I thank you again, Arturo, for this very nice way of finishing the seminars. Please give an applause. And I want to change to Spanish. Como es ya el último día, María y yo queremos agradecer este año de seminarios. Ha sido una labor súper fácil gracias a vosotros. Cómo habéis respondido en ¿cómo se dice? Ofrecer charlas, incluso participar en los seminarios. Es verdad que siempre estaría un poco mejor que estos estudiantes sean un poco más activos, pero estamos trabajando en ello. Y de todas maneras, muchas gracias a Sandra y Carlos por estar siempre ahí, siempre dispuestos a ayudar, siempre a salir de los días. Por favor, un aplauso. Se lo merecen. Y nada, y dejamos el puesto vacante. Es una vacante que es muy fácil de trabajar, ya os digo. Así que esos postdocs que quieran participar, para que no sea la dictadura de María y Javi, para el año que viene hay vacante, ¿vale? Muchas gracias a todos.
Muchísimas gracias.